MK bias. Yes, the bias to from a certain progenitor cells to produce megakaryocyte cells, and it's calculated by the ratio we can see here below in the slide, that is calculated dividing the frequency of a certain clone in the MK cluster and the frequency of this same clone in a non-megakaryocyte compartment. All right. Again, in panel E, we can see these two groups of hematopoietic stem cells that have a low bias and a high bias. Here, a bias to produce megakaryocytes is highlighted in purple, and a greater bias is highlighted in darker purple. All right, and yeah, if cells give rise to multi-lineage cells, it is highlighted in green. And here in panel F, we can see again this single cell map, and we can rapidly identify here hematopoietic stem cells. All right, and they now can see that hematopoietic stem cells have a strong bias to produce megakaryocytes, which is in line with their previous research. Again, in letter G, we can see the distribution of the clones. We can see that well, the important thing here is the hematopoietic stem cells compartment, and they comment that around 30% of hematopoietic stem cells have a strong bias to produce megakaryocytes. And of course, the megakaryocyte progenitors are biased to produce megakaryocytes, and then we can see the non-megakaryocyte progenitors. Then they decided to make a differential expression analysis, and they decided to focus on the megakaryocyte bias subset. And the important thing here is that they identified the same markers. The markers that they identify in this analysis are just the same that they previously identified in the low output group. So here in panel J, we can see that, we can see the four groups of hematopoietic stem cells, and we can see that the cells that are classified as low output are exactly the same classified with the MK bias, and the other way around. Okay. Then in panel K, we can see the correlation values between certain markers of hematopoietic stem cells, and they highlight that the hematopoietic stem cells with low outputs and great MK bias are anti-correlated with the markers of cell cycle score, suggesting that this group of cells are remaining in a quiescent state. All right, which is pretty important. Then in the next panel, we can see these correlation values between the MK bias and the output activity, the information we have already seen, and we can see that the greater the bias, the lower the output activity. All right, so to conclude this figure, the important thing here is that they observed that even after transplantation, a subset of hematopoietic stem cells are remaining in a quiescent state. So now they wanted to see if this subset of cells could reacquire a similar configuration as long-term hematopoietic stem cells that were native. So I think that's all for this figure. Any comments,
Kevin?
till this point?
Kevin? Sorry, there is a question in the chat. How old were the mice in this experiment? I think is 8 weeks. Am I right?
Yeah, they were 8 week old. Both of them were 8 week old. Were the mice from which they extracted the long-term hematopoietic stem cells, and also the ones that were lethally irradiated.
Yep, that is correct.
Any other question?
So Alejo, I wanted to ask if you would prefer to have like the questions at the end, or if anyone in the audience have any questions related to the figures, they could ask through the paper.
Yeah, I mean, it's however you think is more comfortable for you guys. I mean, if the figures are pretty dense and and complex. So if you want to do this like figure by figure or like, you know, that's totally fine.
So I think so as the audience wanted, I would prefer to be at the question and answer session at the end, so we could have like this ongoing of the paper. So thank you. Please go ahead.
Okay, so in the second figure, to determine if any of the transcriptional programs they found before was associated with the capacity of long-term repopulation, they performed secondary transplants. So they repeated the experiment with the barcoding and performed the first transplant, whose clones will be named 1T clones. After a few months, they isolated the cells from the bone marrow and took the half of it to make single-cell RNA-seq analysis, and the other half was split into two and transplanted into two different recipients. After 6 months, they isolated the cells again, and these clones are named 2T clones and analyzed them again by the RNA-seq. So they found that most of the cells, most of the hematopoietic stem cells originated in the first transplant were actually present in the second transplant. And to see whether the cell behavior was random or was influenced by something else, they constructed a distribution over the expected outcomes based on a model where each hematopoietic stem cell behaves independently and has the equal potency to self-renew and to differentiate, almost as if the cells were selected randomly. So additionally, this model takes into account other variables such as the random sampling of the cells, the uncertainty of engraftment of the hematopoietic stem cells, and so on. So this model shows the dependence between these stochastic variables. And here in the panel C, we see that they compare the engraftment potential of the experimental model, which is here shown as blue circles, and the equipotent model, which is a distribution shown here as a rectangle. So, sorry. Okay, so they found a strong correlation of the engraftment potential between the same clones, but in different recipients, by comparing the hematopoietic stem cell compartment, the expansion clones, and also the rest of the kit cells in the compartment. So clearly, the hematopoietic stem cells in different recipients have a higher correlation than the model. And let's remember that this model is assuming that each cell has the equal probability to engraft. So they thought that this high correlation was somehow determined by the primary hematopoietic stem cells. Some properties that were there were influencing the high correlation of the engraftment potential in the cells. So this is a heat map in the panel D, sorry, of clonal frequency, where they determined that the low output hematopoietic stem cells are the most common in the second transplant than the high output hematopoietic stem cells, which is shown in the first part of the figure. And the former cells, the low output cells, maintained quiescent during the first transplant mostly, whereas the latter cells, the high output activity cells, mostly differentiated into the different blood cell types. Is there any question? Or can we go further?
That was beautiful.
Can we go to the next slide, please? Thanks. So using a spring plot to visualize the single data analysis, they found that the clones in the second recipients remained in the compartment of the hematopoietic stem cells in the first transplant. So this result suggests to them that the cells with the capacity to restore the hematopoietic system in the second transplant stayed quiescent during the first transplant. And then by checking the output activity of these cells, it seemed that the cells that had successfully engrafted during the first and the second transplant were low output cells. Thus, whereas the ones who only engrafted in the first transplant had a high output activity. So this suggested that the cell history is somehow determining the repopulation potential of the cells. And finally, they found that the engrafting cells had a low output-like gene expression with genes involved in quiescence such as MIC-N, ProCR, HOXBA, and so on. And there are no cellular cycle genes or genes involved in differentiation. And also, when they compared the data with some databases about native hematopoietic stem cells, they see a strong correlation here. And now with those databases with cell cycle genes or multilineage cells or high output activity cells. So these results in summary indicate that the repopulation potential is heritable, and also it depends on a specific gene expression program that is like those of the native and quiescent hematopoietic stem cells. So we can go to the next slide. Thanks. So it was very logical to think that something was suppressing the output activity of these cells. And by comparing the gene expression of the low output and the secondary repopulating hematopoietic stem cell clones, they selected a bunch of genes that were upregulated and had not been characterized in hematopoietic stem cells before. And they performed a in vitro CRISPR screening to identify the single guide RNAs that increased the generation of progenitor and mature cell fractions. So they transduced the hematopoietic stem cell with the single guide RNA lentiviral library. They transplanted the cells into various recipients, and after the reconstitution, they induced the system by adding doxycycline. Afterwards, they isolated the cells and analyzed them. So they found 5 possible targets that were overexpressed in almost all the populations of the blood cells, as we can see in panel B, and had the highest positive average enrichment score using MAGIC analysis, which is what we can see here in the panel B, from the lowest to the highest score. And MAGIC is basically an algorithm that allows the identification of essential single guide RNAs or genes or pathways from a CRISPR-Cas knockout screen. So among these 5 possible targets, they chose TCF15, which is a transcription factor that was already characterized as essential for pluripotency exit in other cells or was involved in other development of other tissues. So, but it wasn't described in hematopoietic stem cells. So in the presence of the single guide RNA of this transcription factor, when it's not there, the transcription factor, we see in the panel C that there is no enrichment of the hematopoietic stem compartment. And also in the panel D, we can see that if the transcription factor is not present in the differential expression, we see there are not quiescent markers such as the ones that we saw before. So someone is asking, how does the algorithm works? I'm not completely sure. I don't know if Alejo can tell us more about how this works.
Right. I posted the paper in for everyone to see in the chat. I think I mean, I'm not again absolutely an expert in the MAGIC analysis software and how it was written, but I believe that it models basically the distribution of the guides. I think using a negative binomial model, and then it basically, you know, does a test for overrepresentation of the different guides in that distribution, just like any other, you know, statistical test. Yeah, I mean, it's basically telling you whether your guides, in this case for your particular genes, TCF15, are more positively enriched, so on the top side of the distribution, more than expected by this distribution itself. And so it takes into account, obviously, the distribution of all your guides. So the distribution is calculated based on the dispersion of on the values of all your guides that you obtain in the experiment, and then the test is performed. But if you want more details, you can check the manuscript.
Okay, thank you. We can continue, please. Thanks. So also they checked the immunophenotype of the hematopoietic stem cells, where here in this plot, they should be CD48 negative and CD150 positive. And we see that the stem cell population decreases from 21% to 60.7%. And that also is proved with the increase of cells in the different cell cycle phases, as we can see in panel F, when the transcription factor is not there. And also, I think more importantly, is that when they try to make another, the second transplant, the cells without the transcription factor are not able to engraft anymore. So now we can say that TCF15 is altering not only the gene expression involved in quiescence, but is also altering the functionality of the hematopoietic stem cells. Can we go to the next slide? Thank you. So all the way around, when they used lentiviral doxy-inducible TCF15 transgene, they observed that when the transcription factor is overexpressed in the panel H, the quiescent hematopoietic stem cell cluster are the only ones who are overexpressing the transcription factor. And in the panel I, we can see that the enrichment of the is mostly in the compartment of stem cells. When it's overexpressed, the transcription factor is overexpressed. And to see which genes are involved with the transcription factor, they made a differential expression of the cells with the transcription overexpressed, the transcription factor overexpressed versus the control. And they found almost 200 genes involved mostly in quiescence regulation. So these results indicate that the expression of TCF15 is necessary to keep the long-term repopulation capacity in the low output stem cells, and it is essential also to maintain the quiescence of the hematopoietic stem cells.
So once they identify TCF15 as a relevant regulator in hematopoiesis, they ask how the expression of this transcriptional factor is regulated in the native context. And to do that, we use a reporter mouse known as TCF15 Venus. And as we can see here in the FACS plots of panel A and B, Venus fluorescent protein expression was detected a very small percentage of bone marrow cells and was highly enriched in the LT-HSC compartment. And also, in agreement with the single-cell RNA-seq analysis, TCF15 expression within LT-HSC compartment was also markedly heterogeneous. Positive, well, this is not shown here, but also it was positively correlated with surface receptor levels of endothelial protein C receptor and SCA1 that are two markers of quiescent LT-HSC. So we go to the next one. Then to test the functional relevance of TCF15 expression, they separately transplant Venus positive and Venus negative LT-HSCs into irradiated mice, as is shown here in panel D. And they found that venous positive cells constituted blood and bone marrow compartments and regenerated both venous positive and venous negative cells, as we can see in the left part of panel E, whereas venous negative cells almost only give rise to venous negative cells. So the conclusion for that is that there was an impaired primary regeneration and loss of secondary population capacity. And finally, they did a comparison of transplantation efficiency for single or 5 hematopoietic stem cells. They found a frequency of approximately 1 functional HSC for every 2 cells in the TCF15 positive compartment, whereas basically they didn't observe reconstitution activity in the TCF15 negative compartment, as we can see with the black points in the plot of panel F. So in general, the conclusion for this part of the paper is that TCF15 expression defines a hierarchy within hematopoietic stem cells and promotes a self-renewing quiescent TCF15 positive cell state with long-term repopulation potential. And with all this data in mind, they propose a model that we can see in the next slide, where after injury or transplantation, a subset of hematopoietic stem cells loses TCF15 expression and become active and produce progeny. So the next one, please. Right. Before entering the question and answer session, to summarize the main findings of this work, I want to emphasize that this is a great example that shows the power of simultaneous lineage and mRNA profiling. So now we have the possibility to make direct associations of cell behaviors with unique gene expression signatures. In particular, applied to the case of hematopoietic regeneration, they uncover a clone interesting stem cell behaviors and also the molecular mechanisms that regulate them in vivo. On another hand, they propose that TCF15 is one of the few hematopoietic stem cell restricted transcription factors that specifically regulates functional AT-HSC state. And finally, as a perspective, I want to remind that the approach of this study was, may also be directly adapted to investigate stem cell QSN regulators in other regenerative tissues. And it will be very interesting to study in more detail the roles of the other kits found by CRISPR screening to have a more complete vision of the regulation of this process. So thank you all for your attention. And if you have questions, you can ask to the first author.
So thank you very much, Kevin, Valeria, and Victor for this really incredible presentation. So we're now going to start the question and answer session. So please, in order to have like order in the questions, so please write a number in the chat, and and we will give you the opportunity to ask after in consecutive order. So go ahead, and thank you very much, Kevin, Valeria, and Victor for this presentation.
Hello, everyone.
So I'm gonna start with the one I got. Okay, so and who puts the number 1 first? I think nobody. Cecilia. So Cecilia, if you could go ahead.
No, please, you had something to ask. I can be second. No problem.
I don't know who put the number 1.
I think no one put the number 1.
Yeah, so Cecilia, go ahead.
Okay, so first of all, guys, thank you for the presentation. It was awesome. And Alejo, brilliant story as always. Thank you so much. I highly apologize, but I have actually 4 questions and they might be a bit silly. And if so, I apologize in advance. So do you prefer if I ask them all in one go, like the 4 in one go, or 1, then answer 2, answer, or how it's easier?
Go for it, right? I mean,
yes.
If I'll try to respond as briefly as possible to each one.
Okay, great. So the first one, I was thinking whether if you had a look on your data set, if you see stochastic transitions in key regulator genes, meaning if you see transitions, are they stochastic or do they follow a pattern?
I don't think we properly tested for that. It's an interesting question. There's a lot of people looking at those stochastic gene dynamics, but it's something that we haven't done formally. But it's an interesting thing to look at the data. Yeah.
Okay, thanks. Then the second one, I was also thinking that TCF15 is a transcription factor. So I was wondering whether you have done any gene regulatory network analysis using single-cell ATAC-seq or even ChIP-seq for, I don't know, methylation markers or acetylation markers, and if you can really do a sort of regulatory network with TCF15.
Very important point. We're in the middle of doing that right now. But okay. Yeah, I mean, it's a really interesting question. Like, what I can say is that, you know, definitely the genes that we put in the paper, which are these like knockout and overexpression sort of differential gene expression analysis that we've done in figure 3. Yeah, I mean, most of them are not targets of TCF15, sort of as expected. There's a big, probably some reprogramming going on that is just, you know, causing a lot of those genes to go up and down. But the direct targets of TCF15 appear to be very different, actually. What I can say so far is that we're really having to do everything because it's not very well known what is the binding motif of this transcription factor, which makes things harder. So we need to figure that out. It is a VHLH that is known to not really, VHLH transcription factors are always dimerizing in order to drive transcription. We know that it's a transcriptional activator. Those are some papers, I believe, from the Rolls lab from, you know, almost 10 years ago. So it's definitely a transcriptional activator, but what it is dimerizing with, it's still unknown. So we know that by itself, the dimer of TCF15 homodimer is not very active. So it's possible that it dimerizes with other VHLHs. We need to figure out which, and that will also define the motifs in addition to what we know so far. So it's going to be a pretty tough job, but we're trying to do that.
It would be brilliant. Again. Then in line with the TC15 guide, I saw that there are a rare disease named Usher syndrome. And those guys, they have a mutation in this particular gene. I was wondering if you had a look on those guys, if they also show any blood disorder. I couldn't find, I only found death, audition issues, but maybe no one looked at.
Yeah, no, I don't think we've looked at it yet. I know what you're talking about, and I think it's interesting to obviously relate this function. You know, it is true that it is definitely not only expressed in the hematopoietic system. If anything, it's expressed in extremely few cells in the hematopoietic system.
Sure.
So it's widely expressed in endothelial cells as well, which might, you know, as you know, have ramifications in its phenotypes pretty much anywhere. But yeah, it's an interesting thought to just take a look at at some of these parameters in these patients. It's a great idea.
Thank you. And then the last question, it's a bit crazy, and if so, I'm sorry, but I was wondering, is it possible to create like a Larry animal? Like you take ESLs, you put your Larry barcodes, and then you can really follow all the endothelial to hematopoietic transitions and all the YOC-SEC. You kind of have the perfect atlas of hematopoiesis fetal to adult. Would it be this possible?
I mean, it's certainly possible to put barcodes in a similar fashion. In fact, I believe at least the lab of Jonathan Weissman has introduced, I think I'm not sure if it was ESLs, I think it was actually directly to the oocyte, multiple barcodes, and then I'm not sure if it was spontaneous induction or induced, but definitely using a transposon directed approach to try to insert and integrate a bunch of barcodes during early development. And so that approach definitely works. And there's a paper in I think it's in Nature, maybe at the beginning, end of last year, where they put that out. That's, it's possible for sure. And doing it with, Larry barcodes as well, it should be pretty easy. I believe that the most challenging thing is, if I had to do that approach, I would probably do it by modifying the ES cells and then injecting the ES cells with each barcode into the blastocyst. But then the problem is that you still will have, one or two clones maybe contributing to the entire, blood system. Because, I mean, from those numbers of cells that you inject into blastocyst, how many of those clones are going to contribute to the blood system? Maybe 2, 3, 4. It's going to be very challenging. So definitely for those cases, I believe that there's a question somewhere about our more recent model, the Carlin system, which also uses expressible barcodes, but these are generated. The barcode is produced by mutation, by mutagenesis in situ. And those systems are a lot more suited for this type of questions. When you want to label,
point of view?
Yeah, from a processing point of view,
then you have the CRISPR duplications and stuff. Wouldn't be just
it's very hard. No, I'm not going to say that it's not hard. It's definitely hard to use these CRISPR-Cas barcoding systems to reconstruct lineages. It's definitely an unsolved problem mathematically and also I would say biologically. But the systems do work. So I believe that as a sort of technological development, that's going to be highly fruitful moving forward. To continue optimizing and developing better methods to try to use this inducible mutagenesis to generate barcodes in situ. I think that's going to be highly likely where the field will end up moving towards.
Okay, thank you very much.
No problem.
So we have a question from Katarina. It says,
Yeah, I mean, I think that the question is very fitting to what we were just discussing with Cecilia, Larry versus Carlin. I mean, the main advantage is Carlin allows you to generate the barcode in situ. So it doesn't really require you to extract the cells and modify them and then put them back into a mouse. So it's not only beneficial for trying to study, cells in completely unperturbed conditions without removing them from their niche, without perturbing them in any way. And it's also highly beneficial for studying, like Cecilia was suggesting, early development. Perhaps the biggest problem is, like she was referring to, that you don't really control the barcodes that are in your sample. So these are generated every time. And in the Carlin paper, we generated a white list with all the potential or the most likely mutations that are generated by CRISPR-Cas in this model. But obviously the list is not exhaustive. The more cells you sequence, probably, you'll get some more barcodes. And if you look at a different cell population or you modify the parameters of the system in any way, you probably will get some other mutations. So the big problem of the system is that you cannot completely control that aspect. Whereas in the Larry barcoding system, you know how many barcodes you have in your lentiviral library, and you know what are the chances of getting each one of these barcodes into your cell, so you can control for all those parameters, a priori. That being said, I think that moving forward, I believe that in situ barcode generation with these Cas9 systems is going to be the way to move forward, because it allows you to do so many more things.
So thank you. We have so Victor Nieto.
Yes, hi. Congratulations for your work. Also related to Carlin mice, I remember that in that work, you showed that there were differences between different bones. So in this paper, you use whole bone marrow cells. So do you think that if you had taken cells from a single bone, there would be a difference?
For sure. I think that, and this has actually been shown already in very early transplantation experiments when they were tracking, this homing capacity of HSCs through barcoding. It's well known that the process is not perfectly equilibrated across all the bones in the animal, and they're, to try to understand what is driving some specificity or some particular expansion, whether the process of expansion in one bone or another is actually stochastic or not, is something that we definitely thought about pursuing when we started these experiments. We thought about it a lot. But I just decided that it was more important to do like secondary transplants and find whether, which cells sort of maintain this long-term HSC property was a bit more exciting to me. And also for monetary reasons, I have to say, because if you have to process, by single-cell RNA-seq or, each bone separately in many mice, I mean, the costs do ramp up. Now there's better ways of multiplexing things, but when we started this project, the single-cell RNA-seq field itself was still, these droplet methods were still like sort of in their infancy, and there was not a lot of these new fancy tools available, so we couldn't think about a way to multiplex the bones and save up on costs. So at the moment we were like, okay, we just, it's an interesting question, but maybe for the future. But definitely, I mean, it's really interesting to understand whether that distribution across bones is a stochastic process or not. Yeah.
So thank you. So we have Emmanuel.
Yeah. Thank you, Alejo, for being here and for your work. Is very nice. So I want to know if you have more info about the possible, or you have identified the external signal or the signaling pathway that induces the TCF15 expression in these cells.
That's an excellent question. We have not. We know the things that regulate TCF15. Well, I think that one thing that we did discover is that it's probably self-regulating. So if you actually look at the figure 3 and you look at the overexpression model, the differential expressed genes, TCF15, the endogenous copy of TCF15 is actually pretty remarkably there. So to me that says, and that together with the fact that we see that venous positive cells regenerate the venous positive cells, but the negative ones do not. I think that a big important factor defining TCF15, at least expression, is the expression of TCF15 itself. So it's self-regulating as a sort of a positive, feedback loop. But what other things are regulating TCF15 expression in hematopoietic stem cells? I think that's still not very clear. We know that in embryonic stem cells, these are regulated, and many VHLHs are actually regulated by these proteins called ID proteins. So ID proteins sort of bind to this VHLH and sort of inhibit them. And so this is one way that perhaps TCF15 is down-regulated when cells differentiate and become, from an HSC, become a progenitor cell. Maybe ID proteins are playing a role there. And there's also some evidences that this process might be regulated by TGF-beta. And we know that TGF-beta is expressed in the bone marrow, pretty high levels. Actually, it's very important for regulation of HSCs. And so it's highly likely that it plays a role in this process. But I don't actually know the answer to the question yet. It's a really important question that we need to figure out.
Yeah, maybe it will be a perspective for your future work.
It's certainly a possibility.
Thank you.
Thank you so much. It's a great question.
Okay, so we have Maria Jose.
Yeah. Hi. Hi, Alejo. Thank you very much for being here. I had a question about, because you showed that TCF15 regulates quiescent factors in the HACs, and I was wondering what could be next? I mean, what could be done regulating TCF15 to allow the cells to re-enter the cell cycle? And I was thinking about your single cell RNA-seq analysis, where you compare the low output HECs versus the high output. Did you find any factor there that could be explaining this, like downregulating TCF15?
Yeah, short answer, I don't know. Definitely, it's interesting because the fact that the property of self-renewal, the way that we find it, seems to be highly intrinsic, right? Would suggest that whatever braking mechanisms are sort of already a bit, more expressed in these clones that self-renew and do not differentiate, and do not depend too much on, necessarily on these extrinsic effects, right? But then certainly they have to have the chance to do it, because if you do the serial transplantation, you can see those clones reactivate and become now, more active and rescue the hematopoietic process in the secondary recipient, right? So then the question is, what causes that transition to happen, and whether that is perhaps something that occurs, during the early process of transplantation, some sort of may depend on actually, like you say, cell cycle signals. I mean, we really don't know. I don't think that it's going to be an intrinsic sort of completely self-regulated model. I think that definitely there are some cues from the bone marrow. We know that there's a lot of cytokines that are very important that are produced during these phases, KIT ligands and colony stimulating factors that are produced during the, there's a big actual, increase in the concentration of cytokines when you irradiate the bone marrow. And so for sure, a lot of those will play a role. So some extrinsic signals will play a role. But I think that it is possible, like you suggest, that there is some data hiding in this analysis that would sort of guide our efforts to finding which pathways are more important. So yeah, I mean, we have to do that work, but it's an interesting question. It's also a very complicated problem, definitely, yeah.
Thank you. And you just sort of answered my second question, which was if there are any niche factors important. So I'm gonna flip it and ask you, is it possible to track for each of the subset of HCs that you showed, for example, the high output and the low output, where especially they are in the bone marrow, like whether if they're very close to each other and signaling to each other, or something like that?
I would love to know the answer. That's a really cool question. I suspect that actually a lot of the intrinsic program, and someone was talking about the, I don't remember if it was Victor or I think it was Victor, but at the end of the journal club, he was suggesting that it would be interesting to look at what the other genes in the screening, the hits identified in the screening, what they are doing. Actually, if you look at the list of genes that are expressed in these serial engrafting cells, a lot of them are extracellular molecules and receptors. And I suspect that a big part of this intrinsic sort of stem cell memory is linked to some sort of autocrine regulation or perhaps a self-regulated mechanism to find a particular niche in the bone marrow. And so that could probably play a role. I don't know if they recruit the cells themselves, they stimulate their own, they create their own niche directly, they attract other cells to them, or what is exactly the mechanism. But there's definitely that possibility that localization plays a big role and niche components. But it's cool that it's probably intrinsically regulated.
Thank you very much.
Yep. Thank you. Great.
So thank you, Majo. So I got a couple of questions too, Alejo. First of all, congratulations and thank you very much for accepting to join this session. So my question is more related with the ones I have previously shown. So I was looking at that in 2013, they showed that fibroblast growth factor was in fact the signal that induced expression of TCF15. And I was looking at some data on hematopoietic stem cells and fibroblast growth factor. In fact, the knockout receptor of the fibroblast growth factor in a hematopoietic stem cells resembles much of the data that you are showing here in the paper. So I don't know if fibroblast growth factor, in fact, is one of the key players of the signaling in this long-term hematopoietic stem cell. In fact, just one more point, I was looking that also fibroblast growth factor actually signaling through the megakaryocyte formation. So maybe could be that pathway. I don't know. What do you think about that?
Definitely. I mean, obviously it's hard to disentangle the whole thing unless we really know what are the sort of directly linked components in this pathway, right? At this point, like you suggest, we know about a series of receptors and ligands and cytokines that can regulate this protein and this process. And there's a lot of things that, like you suggest, like TGF-beta, regulation is required for maintaining HSC quiescence, FGF receptors. There's, I believe, 1 or 2 receptors for FGF that are expressed in HSCs actually quite specifically, and we even find them in our some of our analysis.
Yes,
and they could be playing a role there. The question is, the problem is a lot of these knockouts, right? A lot of these receptors and pathways are not super specific of just HSCs, right? So when you create a knockout, even if it's a blood-specific knockout, you will have this mutation in a bunch of cells, right? And then you don't know if it's like, something that some other cell is not producing, or it's a direct or indirect effect, and so on. And so we need to figure out a way to perhaps alter or perturb these pathways specifically in stem cells, or maybe to try to start connecting the dots. So, more directly, like what are the things that induce TCF15? Now that we have this reporter mouse, it's going to be probably very easy to try to figure out, do a quick screening with a bunch of cytokines or a bunch of, even CRISPR screenings to try to identify what things regulate TCF15 expression and so on. So I believe that it's, it's highly relevant to look at some of these pathways that have already been published, and it's easy to try to connect them. But to properly delineate the signaling pathway, we need a lot of biochemical work first. Yeah.
So I got just 2 more questions related to expression of TCF15 in aging, because we know that in aging, like the biology of long-term hematopoietic stem cells actually changed towards like a more myeloid lineage. So I don't know if you're planning to look, and any interaction with GATA3 as was one of the first factors like that showed to be like related with long-term hematopoietic stem cells.
It's definitely possible that GATA factors might be involved. In fact, there's a strong correlation we've done a little bit of ATAC-seq and other sort of chromatin regulatory landscape type experiments with these TCF15 cells, and it looks like GATA definitely will be part of the picture. That's at least how it looks right now. We haven't detected an interaction or a complex yet, but other VHLHs have been reported to do this in a bigger complex with GATA, so it's not unlikely that we'll find something. Related to the first part of the question, I think that, well, we have some data that during aging, the expression per cell is actually not changing. So the expression of TCF15 itself per cell is not going higher. We do know that the number of cells that have this sort of self-renewing low output behavior actually continue to self-renew and sort of expand during your lifetime. And in fact, some of them fall into probably some epigenetic differentiation traps, and they cannot actually differentiate very efficiently. So whether that whole process, is somehow related to TCF15, we don't know. But what I can tell you is that the percentage of venous positive cells is going higher with time. So that percentage, that group of self-renewing cells that are low output and sort of quiescent and dormant and self-renewing but not differentiating, which goes higher with age, they all express TCF15.
Yes, because I was looking that there are some data that very interesting that long-term hematopoietic stem cells actually count their cycling. So maybe they will be enriched. I don't know. I'm thinking about TCF. It's really interesting. Yes.
Totally possible. Totally possible what you say. Yeah.
Thank you very much, Alejo. So
thank you. Thank you.
We have another question from Neil Harris. He says, are the leukemia patients with mutations in the TCF15 gene?
Not that I know so far, at least.
So Aranza Su asks if there are those mice germ-free, if there like any relation with microbiota.
No, great question, and there's a lot of links between microbiota and HSCs, and there's a lot of interest in that field. Definitely interested myself in trying to understand that process more with actually using Larry for trying to understand how microbiota might and just sensing of some of these antigens can actually end up reprogramming the memory of stem cells, and use Larry for that. I have quite a few projects that I'm planning to do when I start my own lab with that. But I think that connection to TCF15 unknown. And no, the mice in the experiments were not germ-free.
So thank you. So Valeria?
Yeah, hi. So thanks for being here, Alejo. I wanted to ask you if you think that this expression of TCF15 is like is caused by the transplantation per se, or is it present in like in normal conditions, or if you think that there is another pathway or in the native hematopoietic stem cells, and this is only because of the transplant experiments.
To completely answer that question, we have to do the knockouts and everything in the completely unperturbed native system, right? And to do that, we need to generate the new mutant, because unfortunately, the one that exists right now is a knockout that's like total knockout, full body knockout. And unfortunately, the mice do not make it during development. So the problem is this TCF15 transcription factor is really important for somatogenesis. And if you don't generate your muscles normally, you end up having a lot of problems. And obviously the fetuses actually of the embryos die. So to try to do that, we've generated recently a conditional knockout. And we're trying to do those experiments in a completely unperturbed setting to see what is the, to what extent these phenotypes are linked to transplantation and so on. And only in transplantation, like you suggest, which is possible since, these cells are, these HSCs are more likely, not highly active, not producing progeny. Getting rid of them, a mouse would not really notice them in principle. That's our idea, that's our hypothesis. The mice are probably going to be fine for a long time until probably they have an infection or some sort of perturbation and so on. They probably should be fine without these cells. And then maybe this is a sort of transplant specific or injury specific regenerative response that requires these cells, right? But definitely I expect that TCF15 will be necessary to maintain them in this state in situ. But that's, we like you suggest, we need to test that formally. What I can tell you is that the expression of TCF15 itself, we've looked at that in a completely unperturbed, this is a completely native system. That's a reporter knocking in the endogenous TCF15 allele. And so that is reporting the endogenous expression of TCF15. So the specific expression of this transcription factor, for sure, that's there without any transplantation conditions.
Thank you.
So we have more questions in the chat. Rafael asked could there be a spatial relationship between low output HCS and megakaryocyte-based HCS cells inside the bone marrow? Could niche factors molecules also regulate the stem cell behavior of hematopoietic stem cells?
Yeah, I think we've touched upon those topics a little bit. Definitely, there could be. I'm probably more leaning towards the fact that these two populations, the megakaryocyte biased HSCs and low output HSCs, might actually be very similar and even localized very similarly. The question is whether the high output ones, which are able to, self-renew because they remain there for many months, but they are producing progeny, are those localized differently? And that's something that we still need to figure out. But yeah, really important question.
So you have your results show nicely that TCF regulates factor of quiescence, how is the exit of quiescence and interest of different regulated afterwards.
Yeah, I mean, I think we've also discussed that a little bit.
Yeah.
The exit of quiescence is a really important problem, and we still don't know many aspects of it. We do know that there's a series of, like you probably expect, growth factors, signaling pathways that end up regulating the expression of cycling dependent kinases. There's one that is very important, CDK6, and that drives sort of part of the process. We know that MYC transcription factors also play a massive role in this, and so the upregulation of the expression of MYC transcription factor is actually sufficient also to drive this process and required to drive this process. There's an entire literature that we could spend another hour revisiting, but there's a lot of pathways that play into this connected to TCF15, none that I have seen yet. That's sort of the important point. Like, we still need to try to understand how it's this transcription factor is connected to all of these other observations.
Okay, so I think we will wait for 2 more questions. So if you could choose anyone in the chat, and then if anyone would like to contact Alejo to further go to this discussion. So any question you find interesting?
Yeah, I'll try to quickly go through a few of them. Which factors downregulate? So Caleb is asking which factors downregulate TCF to allow cell cycle entrance. I think it's a very similar question to the previous one. No candidates yet, apart from things that are already being published, and you can look those up, but in the paper. If you can multiply the HSCs by activating the progenitors, I am not sure what Kinley means by multiplying the HSCs. What I can tell you is overexpression of this TCF15 transcription factor does not actually increase the number of HSCs. It just sort of really keeps them quiet. It caught my attention. Soaf asks that there are fatty acid oxidation enzymes. Those have been published actually quite extensively in the literature. Fatty acid oxidation regulation is definitely important for the process. Why and how it's related to chromatin modifications, which is really the really exciting question that is linked there. Definitely an interesting axis to explore. Maybe not my area of expertise, but I would say that there's a lot of interest in that area from many labs, and I expect that people will pursue that to some extent. But yeah, really cool question there. So Agnieszka is asking, within the network of transcription factors that TCF15 regulates, is there anything that promotes metabolism activation in these cells? I mean, the question by Zoff is kind of along those lines. In terms of as a transcriptional regulation, how this whole metabolic program is being reprogrammed, not clear yet. But you can, the list of genes are published. You can mine them yourselves and take a look at if anything sort of rings a bell for you guys. Not a metabolism expert myself. I do tell you that I believe FOXO transcription factors were part of the overexpression signature. So maybe FOXO, mTOR, I believe pathway genes were also present in the overexpression signature. So those might be maybe related. Do you think that this could lead to an evolutionary advantage in mammals? Does this occur in all species? I have no idea. I have looked at humans. Definitely there's some expression of TCF15 in human HSCs as well. Not sure about other mammals, unclear about other vertebrates or below that. And finally, Carla is saying, do you think that overexpression of TCF15 in a small group of HSCs would be useful on clinical transplants? Well, definitely, if I am going to get a transplant, I want my cells to be expressing TCF15, that I know. Whether there is a way that we can use this system to try to make more healthy HSCs, definitely that's super viable. You just take the HSCs from with these reporter mice, and then you try conditions that optimize the frequency of these cells and the percentage of these cells. That should lead you instantly to some sort of screening for drugs that optimize the transplantation process. I suspect that should not be too hard to do, but definitely, it will take some time to set up those big scale experiments, but something that someone should definitely follow up on.
So thank you very much, Alejo. Just before we leave, we have like this tradition. Could you like tell the audience like the take-home message of your paper, like the message you want everybody when you read the paper, like they know, okay, this is the take-home message I should take.
For sure, yeah, it would be my pleasure. First of all, thank you all so much for being here and for doing this. I think it's really cool. Started following you guys on Twitter very recently after my colleague Sarah, I believe, presented the Carlin paper not too long ago. And I think it's a fantastic thing that you're doing, and I'm really happy that this is continuing, and obviously honored to be part of the first PJC developmental biology. That's really cool. And the main message, I think that the number one thing that we found out is that, using this type of barcoding technologies, you can actually follow up clones for a really long time. In our cases, 4 months after we had transplanted the cells. And you can see that there are some behaviors that are sort of memorized in the clone. And we don't really know how that process is happening. And it looks like the capacity of these clones to maintain the serial transplantation potential is definitely one of those properties that is memorized and probably even set on those cells, even very early during development. We still don't know how early that memory is established, but it perdures. It's maintained over time. And the cool thing is that if you use these barcodes combined with single cell lineage and RNA analysis, like we did with Larry, you can actually compare these clones and see whether some genes are predicting the behavior or correlating, at least correlating with the behavior of the clones. And if you think about it, it's, you have, these type of experiments, when you have these sort of fates that are memorized in a clone, the genes that you reveal by doing this analysis is almost like a functional genomics assay, because genes that are a bit more highly expressed in one population versus in one clone versus another are linked directly towards the function of that clone, right? So it's almost like you did like a knockout screening without doing any sort of knockouts, because you know that slightly higher levels of these and these and these other genes are linked to this particular functional behavior, and they're linked more than just by chance. So I think that's the most important thing. Just outside of hematopoiesis, outside of blood development and HSCs, just think about how you can use these tools to unveil this type of memory behaviors anywhere and actually compare the behavior of clones and the expression levels of these different clones. You can use this in cancer. You can use this in immunology for different responses by immune cells. There's really a plethora of potential systems where this could be highly beneficial. So regeneration, again, it's just one of them. Really, it's really any behavior that you can think of where you have a population of cells that is expanding or self-renewing and so on, where you can do this clonal tracing, you can do this. And so, yeah. Anyhow, I think that has to be the sort of clearer message here beyond the model system itself.
Thank you very much, Alejo. Just before we go, can we take a picture of all of the meeting? So anyone in the audience, if you could turn on your camera. And anyone in the team, if they could help me taking also the picture so we could take anyone in the audience. So hold on a second. So I will be taking the pictures. And so once again, thank you everyone for being here. And please follow our social media to more updates on Public Journal Club and Public Journal Club Developmental Biology. So Alejo, once again, thank you very much. And we will look forward to your research, incredible research, and congrats on your new lab.
Thank you.
Bye.
Thank you.
Thank you, Alejo.
Thank you, guys. Thank you so much.
Okay, so here. Hold on. Okay, so, gracias, Kevin. Gracias, Valeria, Víctor. Muchas gracias. Súper chido. Entonces, espérense, déjenme.
Gracias.
Voy a cancelar la sesión y ahorita nos conectamos.
Se fue.
Sí. Voy a cancelar la sesión y hablamos.
¿Ahorita nos conectamos?
Ajá, sí.
Pero en este
¿Nos
mismo
reconectamos? Ajá.
¿O quieres que haga uno de Google Meets o
Creo que sí, Google Meet estaría bien. Espera.
quieres que lo haga?
Sí, please.
Va.
Gracias. Ya entró una, o sea.
Sí, ya vi. ¿Qué onda? Admití.